In our next lesson on Chapter 8, Lipids and Membranes, we want to look at the composition of the bilayer and lipid movement. What we find is that the lipids move within the bilayer, that is, laterally, very easily, although there are some limitations, as we'll see in the next lesson. But movement between the monolayers, which involves translocation, is much less frequent. The question is, why is that? Let's think about what's involved in that process. In order to move a lipid molecule from one side of the membrane to the other, I have to move that polar head group through a very nonpolar environment to get it to the other side. Clearly, this would be a non-spontaneous process because we're moving a polar molecule into a very nonpolar environment. This can depend somewhat on the type of lipid as far as the ease with which this is done. Usually it requires a protein or enzyme for this translocation, and they're called generally translocases. But we'll find that the movement of lipids from one side of the membrane to the other, from one monolayer to the other, is actually essential for maintaining the asymmetry and function of the bilayer. So we find that the two leaflets, the two monolayers in the bilayer, are not identical. There is an asymmetry, and that's well as illustrated in our table here. We'll notice that phosphatidylethanolamine is usually localized to the inner monolayer, whereas phosphatidylcholine is usually in the outer monolayer. And you can see it varies quite a bit depending on the lipid, but they do tend to localize on one side or the other rather than being equally distributed between the two monolayers. Some of this has to do with the orientation of the enzymes that make the lipids, which side of the membrane they face, but also some of this has to do with these translocases or flipases that move them from one side to the other. It's not important that you remember this table or which lipid faces which side of the membrane, just that there is this asymmetry. It's a clue to us that the function of the membrane is not just as a cell sac. It's not just a structure. It's playing a more direct biological, even a chemical role, as we'll see. In our next video lesson, we want to see how proteins associate with membranes and see if there are some common structural motifs for these membrane proteins.